Children, today we are going to start with the topic protein synthesis. Protein is very much important part of our cells, and how protein is synthesized, that we have to see in this topic. So before starting protein synthesis procedure, we have to see the structure of cell. Everybody knows that outermost covering of the cell is the cell wall. Inside the cell, cytoplasm is there, and in that cytoplasm small organelles are also present, and very much important, nucleus is also present. Okay, and inside the nucleus chromosomes are present. Chromosomes are made up of DNA. DNA means DNA is present inside the nucleus, and RNA is present in the cytoplasm. RNAs are of three types. First one, it is messenger RNA, or it is also called as mRNA. Messenger RNA, it is temporary. Then next is rRNA, it is called as ribosomal RNA. And third one is tRNA, that is transfer RNA. So these three different types of RNAs are present. Now here, from DNA to messenger RNA, DNA itself prepares messenger RNA inside the nucleus, and rRNA and tRNA are present in the cytoplasm. So formation of mRNA from DNA, this procedure is called as transcription. What it called as? Transcription. Now after completion of this transcription process, means preparation of mRNA, this mRNA comes out in the cytoplasm. Whenever it will come out of the cytoplasm, from this mRNA protein is synthesized, and this procedure is called as translation. This procedure is called as translation. Means mRNA is formed from DNA is called as transcription. This mRNA comes in the cytoplasm, and it from this mRNA protein is synthesized. This procedure is called as translation. Translation takes place in the cytoplasm. Transcription takes place inside the nucleus, and together transcription and translation together called as central dogma. Central dogma. Two procedures, transcription and translation together, called as central dogma. Now this is, in short, we have seen how protein is synthesized from DNA, but now in detail we have to see how protein is prepared or formed from the DNA. Now for that, first we have to revise the structure of DNA. Last year in 9th standard we have already seen the structure of DNA. It is a double-stranded structure. We know that DNA means what? Deoxyribonucleic acid, and it is double-stranded structure. And RNA means what? Ribonucleic acid, it is single-stranded structure. Now we know that DNA is double-stranded structure. It is ladder-like structure which is twisted around itself. Last year we have already seen. One more important thing we have learnt last year, that DNA is made up of 4 different nitrogenous bases. First one, adenine. Second one, thymine. Third one, guanine. And fourth one, cytosine. 4 different nitrogenous bases are present, along with deoxyribose sugar and phosphate molecules are also present in this DNA molecule. One more important thing we have learned, always adenine paired with thymine, and guanine paired with cytosine. This also we have seen. So we have revised the structure of DNA. Now how transcription takes place, that we have to see. See, for example, this part is required as a template. So this part will unwind. Remaining part is same. Remaining part is winded, but only this much part is unwind, and you can see nitrogenous bases on this side. A is paired with T, C is paired with G, like that pairing is already there. Now it is unwind and new mRNA is formed inside it. New strand is formed inside it. But remember that in DNA thymine is present. Instead of that, in RNA uracil is present. What? Uracil. Instead of thymine, uracil is present in case of RNA. So here you can see wherever this new strand is formed, in that case always A is paired with U. You can see this is A. It is always paired with U. Instead of T, everywhere U is there. So this is the new strand form. It comes out in the cytoplasm. This procedure is called as transcription, preparation of mRNA. This is the mRNA. Now it comes from nucleus to cytoplasm. This is the strand of mRNA. Now here you can see different nitrogenous bases on it. Okay, so these different nitrogenous bases you can see, and now procedure of translation is going to start. Before that, one important thing we have to keep it in mind. On this mRNA, number of nitrogenous bases are arranged, but AGC is the codons. Those are called as starting codons. See here, the mRNA bases are there. They are arranged in the pairs of 3. So those pairs are called as codons. These are the codons. 3 pairs they have made. They are called as codons. So this is the first codon. It is always AGC. It is called as starting codon. Starting codon. So number of codons are present on this mRNA, but wherever AGC codon is there, from that place it will start transcribing proteins. Now here what will happen? Now transfer RNA along with mRNA will come here, having 3 complementary codons. For example, for A, which is complementary, U. For G, C is the complementary codon, and for C, G is the complementary codon. So it will bring these complementary codons called as anticodons. Anticodons. So this transfer RNA will bring complementary codons, and with that specific type of amino acid will also it will bring. After bringing that specific amino acid, this transfer RNA will leave this place, leaving this amino acid here only. Then next transfer RNA will come. It will bring the anticodons, set of anticodons. Complementary to U is A, G is C, and again for A complementary is U. So these anticodons it will bring, and with this set of codons one specific type of amino acid it will bring. Let us consider this is AA, this is AA1. Then this transfer RNA will leave the place, leaving this amino acid here only. Again third transfer RNA will come, it will bring codons, complementary codons, and again for these complementary codons specific type of amino acid it will bring. Again it will leave the place and amino acid will be here only. In this same way number of amino acid will collect over here, and these amino acids will join with each other with the help of bond. So this transfer RNA will leave this place and this chain of amino acid will form here in this way, and this chain of amino acid is called as protein. So protein is formed, and this procedure is called as translation. This procedure is called as translation. So formation of this chain of amino acids is from messenger RNA is called as translation. After formation of this chain, the mRNA destroyed or it vanishes, and that's why here we have said that messenger RNA is temporary. After this procedure of protein synthesis, this messenger RNA vanishes, finishes, and that's why it is called as temporary RNA. So here we have seen in this topic, we have seen how transcription takes place, how mRNA is formed from DNA inside the nucleus, how a protein is synthesized from mRNA inside in the cytoplasm, means process of transcription we have seen in detail, process of translocation also we have seen, and together it is called as central dogma. There are 20 amino acids present in the nature, and out of using those 20 amino acids, combination of amino acids takes place, and thousands of different types of proteins they can prepare. So according to the requirement, cell prepares the specific type of proteins using DNA present inside the cell. So I think you understood the procedure of protein synthesis. Thank you so much.